Hello everyone. In this lecture, we'll talk about the metabolism of proteins and amino acids. But before we proceed, here is a very quick revision. We have learned that in the station of metabolism, we have the breakdown of macromolecules such as proteins, carbohydrates, and fats into their respective building blocks, such as amino acids, monosaccharides, and glycerol with fatty acids. So all these building blocks will eventually be converted into the 2-carbon acetyl coenzyme A, which is the universal substrate that will go into the stage 3 of metabolism, by which you will get ATP production. So there are two steps with the conversion of a monosaccharide into acetyl coenzyme A. So what are these two processes? Well, the first one is glycolysis, which involves the conversion of the 6-carbon glucose molecule into the 3-carbon pyruvate molecule. While the second reaction is oxidative decarboxylation, where we convert the 3-carbon pyruvate into the 2-carbon acetyl coenzyme A. Now, how about fatty acids? Well, fatty acids can be converted into acetyl coenzyme A by a process known as beta oxidation. While for our lecture today, our focus will be on both proteins and amino acids. So how do we get energy from amino acids, and how do we deal with the waste product coming from the breakdown of amino acids is the question we're trying to answer today. Okay, this is the basic structure of an amino acid. You can see that it is linked with 4 different chemical groups, which are the carboxyl group, the amino group, the side chain R, which gives the amino acid its specific chemical and physical properties, and a 4th hydrogen atom. So we know that these amino acids are just monomers because they can actually be involved in polymerization with the use of the peptide bond to form a long chain of polypeptide. So the polypeptide is a very strong structure because the peptide bond is essentially a covalent bond that is having a very high bond energy, and therefore it is very strong. And depending on the type of R group you have, the amino acids could be either non-polar, which means there is no net charges and the side chain is hydrophobic, or it could be positively charged when you have a net positive charge. So you could see that in the example of lysine, because you have 2 positive charges with 1 negative charge, so that gives you a net charge of positive 1. Or it could be polar, such as in the case of serine, threonine, and tyrosine, where you have the hydrophilic side chains that comes with an OH group. Or sometimes the amino acids could be negatively charged, which means again the net charge is negative. So here's one important point to take note is that when we say an amino acid is charged, neutral, or polar, we're referring those charges or those state of ionizations at pH 7.4, which is our physiological pH, because at different pH you can get different charges on the amino acids.